Hello students, welcome back to my channel. Today I am discussing exercise of the chapter 11 photosynthesis in higher plants from NCERT book, as you can see it over here. See the first question, by looking at a plant externally, can you tell whether a plant is C3 or C4? Why and how? The correct answer is here. See, by looking at a plant externally, we cannot tell whether a plant is C3 or C4. Unlike C3 plants, the leaves of C4 plants have a special anatomy called Crane's anatomy, and this difference can only be observed at the cellular level. For example, although wheat and maize are grasses, wheat is C3 plant while maize is C4 plant, as it has the Crane's anatomy. Your second question is, by looking at which internal structure of a plant can you tell whether a plant is C3 or C4? Explain. See, we can determine whether a plant is C3 or C4 by observing the cross section of its leaf under a microscope. C4 plants possess Crane's anatomy, where vascular bundles are surrounded by a distinct ring-like layer of large bundle sheath cell, whereas in C4 plant, bundle sheath cells, the vascular bundles are surrounded by a ring-like layer of large thick-walled parenchyma cells which are rich in chloroplast, and they contain two types of chloroplast, granule in mesophyll cells and agranule in bundle sheath cells, and no intercellular spaces are present, and the bundle sheath cells are tightly packed around the vascular bundle with almost no intercellular spaces. Whereas in case of C3 plants, they lack Crane's anatomy. The vascular bundles are not surrounded by such distinct chloroplast and chlorophyll-rich bundle sheath cells. Photosynthesis occurs only in the mesophyll cells. By observing these structural cellular level features in a leaf cross-section, we can distinguish or differentiate between C3 and C4 plants, right? Now the third question, even though a very few cells in a C4 plants carry out the biosynthetic Calvin pathway, yet they are highly productive. Can you discuss why? Your answer is C4 plants are highly productive, though few cells performing the Calvin cycle only in bundle sheath cell, because they possess a specialized Crane's anatomy that drives a highly efficient carbon dioxide concentration mechanism. This mechanism using PEP, that is phosphoenol pyruvate carboxylase in mesophyll cells to deliver carbon dioxide to bundle sheath cell. It minimizes the photorespiration and maximizes Calvin cycle efficiency, and the main reasons for their high productivity include suppression of photorespiration, means photorespiration is very low or it's totally absent, rapid carbon dioxide delivery, adaptation to high temperatures, and efficient carbon assimilation. I hope it is clear. Fourth question, ribulose bisulfate carboxylase oxygenase is an enzyme that acts both as a carboxylase and oxygenase. Why do you think that ribulose bisulfate carboxylase oxygenase carries out more carboxylation in C4 plant? See, ribulose bisulfate carboxylase oxygenase carries out more carboxylation in C4 plants because of Crane's anatomy. In C4 plants, ribulose bisulfate carboxylase oxygenase is not present in the mesophyll cells, where initial carbon dioxide fixation occurs. It is exclusively located in the bundle sheath cell. Carbon dioxide concentration mechanism. In this, the mesophyll cells first fix carbon dioxide using phosphoenol pyruvate carboxylase, which has no oxygenase activity, to form a 4-carbon acid like malic acid or aspartic acid. This is transported to the bundle sheath cells, where it is broken down to release carbon dioxide. So this mechanism creates a high concentration of carbon dioxide directly around ribulose bisulfate carboxylase oxygenase, and lack of photorespiration means because photorespiration is generally absent, or if it is there, very low, and high affinity for carbon. Ribulose bisulfate carboxylase oxygenase has a higher affinity for carbon dioxide when it is abundant, and the high carbon dioxide environment ensures it acts primarily as a carboxylase. Therefore, C4 plant use specialized anatomy to flood the ribulose bisulfate carboxylase oxygenase enzyme with carbon dioxide, maximizing its efficiency. The fifth question: suppose there were plants that had a high concentration of chlorophyll B but lacked chlorophyll A. Would it carry out photosynthesis? Then why do plants have chlorophyll B and other necessary pigments? Your answer is, a plant with high chlorophyll B but lacking chlorophyll A cannot carry out photosynthesis, because chlorophyll A is essential, because it forms the main reaction centers of photosystem 1 and photosystem 2, converting light energy into chemical energy. Without chlorophyll A, the energy captured by chlorophyll B cannot be utilized. Plants have chlorophyll B and other pigments as chlorophyll B carotenoids and xanthophylls function as accessory pigment. They absorb light at different wavelengths, means at the low wavelength, than chlorophyll A, and transfer this energy to chlorophyll A, and they protect chlorophyll A from photooxidation, which can damage the photosynthetic machinery. So chlorophyll A is essential for photosynthesis, while chlorophyll B and other acts as a helper pigments to increase efficiency and safety. Your next question: why is the color of a leaf kept in the dark, frequently yellow or pale green? Which pigment do you think is more stable? A leaf kept in the dark turns yellow or pale green because the absence of light stops the production of chlorophyll, the green pigment essential for photosynthesis, causing it to break down. Without chlorophyll, more stable pigments like xanthophylls and carotenoids becomes visible, and chlorophyll production is directly dependent on light. So when removed from light, chlorophyll degrades. Xanthophylls and carotenoid pigments are more stable and do not require light for their existence, making them visible when the green pigment disappears. Next question: look at leaves of the same plant on the shady side and compare it with the leaves on the sunny side, or compare plant the potted plants kept in sunlight with those in the shade. Which of them has leaves that are darker green? Why? मतलब ठंड में जो छाया में है पेड़, उसके लीव्स ज्यादा डार्क क्यों होती है? See, the increased concentration of chlorophyll in shaded leaves make them appear darker green to compensate for the lower intensity of sunlight. On the other side, leaves in direct sunlight receive abundant, means more energy, means they do not need as much chlorophyll, so resulting in the lighter green color. I hope it's clear. Next question, in ये क्वेश्चन नंबर 8, इस क्वेश्चन नंबर 8 में देखिए आपकी ये दिया हुआ है, figure 11.10 shows that effect of light on the rate of photosynthesis. Based on the graph, answer, write the question. Your graph is this one. This is the graph, like 11.10 graph. See here in the intensity, when you see, now you see your the first option is question is here. Here, at which point A or B or C in the curve, a light is the limiting factor. Your answer say in the reason A, light is the limiting factor. Now the other, what could be the limiting factors in the reason A? अब reason A में all limiting factors क्या हो सकते हैं? The other limiting factors in this region, they can be temperature, carbon dioxide and water. Now here, what do C and D represent on the curve? Now here you can see C represents constant rate of photosynthesis and D represents constant light energy, right? Now the last question, give comparison between the following. Comparison between the first C3 pathway and C4 pathway. See, C3 pathway, first stable product is 3-phosphoglyceric acid. Here first stable product is oxaloacetate. In C3 pathway, second point, primary acceptor is ribulose biphosphate, RUBP. Here primary acceptor phosphoenol pyruvate, that is PEP. Here fixation enzyme is ribulose bisulfate carboxylase oxygenase only. Here fixation enzyme is phosphoenol pyruvate, that is PEP. Fourth point, photorespiration is high. Here photorespiration is very low or it is absent totally. Here it occurs in the mesophyll cells. Here it occurs in mesophyll and bundle sheath cells in case of C4 pathway. They have Crane's anatomy is absent in C3 pathway, whereas in C4 pathway, Crane's anatomy is present in case of C4 pathway. Your next difference, cyclic photophosphorylation and non-cyclic photophosphorylation. See, in cyclic photophosphorylation, it is related with photosystem 1. It is associated with the photosystem 1 and photosystem 2. Cyclic, the electron released from chlorophyll molecule is cycled back. Here the electrons are not cycled back but compensated by the electrons from photolysis of water. Here photolysis of water does not occur and oxygen is not evolved. Here photolysis of water and evolution of oxygen occurs. Fourth point, photophosphorylation occurs at the two places. Here photophosphorylation takes place only at one place. Here NADP is not reduced. Here NADP plus, this plus sign is reduced to NADPH2. Your last difference, anatomy of leaves C3 plants and C4 plant. Now see, here leaves do not possess Crane's anatomy, C3 make Crane's anatomy. Here the leaves have Crane's anatomy. Chloroplasts do not have peripheral reticulum. Chloroplasts, they have peripheral reticulum. Chloroplasts are of one type and is monomorphic. एक ही तरह के होते हैं. Here there are two types chloroplasts, they are dimorphic. Bundle sheath cells usually do not contain, इसमें bundle sheath cells में chloroplast नहीं होते. Here bundle sheath cells possess prominent chloroplast. Here chloroplasts are granule, chloroplast में granule होते हैं. Here chloroplasts are agranule in bundle sheath. Bundle sheath के chloroplasts जो होते हैं वो agranule होते हैं. In C3 plant, mesophyll cells perform complete photosynthesis. Here mesophyll cells perform only initial fixation. Here perform photosynthesis only when stomata are open. Here perform photosynthesis even when stomata are closed, that is from the CO2 produced in respiration. देखिए C3 प्लांट्स में क्या होता है, ये स्टोमेटा है, इनमें क्लोरोप्लास्ट नहीं होते हैं, सिंगल लेयर वास्कुलर बंडल, सिंगल लेयर्ड बेंचेस. यहाँ पर जो क्या होता है, ये बंडल शीट होती है, इसमें क्लोरोप्लास्ट C4 प्लांट्स में होते हैं और इनमें क्लोरोप्लास्ट प्रेजेंट होते हैं, बंडल शीट टाइटली क्लोज। ये जो होते हैं, इसमें जो ये स्पेस मैंने बनाए, ये नहीं, ये पूरे कवर्ड होते हैं, इसमें बिल्कुल भी इंटरसेलुलर स्पेसेस नहीं होते हैं। That is what the basic difference between C3 and C4 plant. I hope everything is clear. Any doubt anywhere, you can ask me in the comment section. Please subscribe to my channel. Again, we will meet in the next video. Thank you.